Hi. There is a saying that life and time are the world's best teachers. Life teaches us to make use of time, while time teaches us the value of life. So we all should try to utilize our time wisely and value the life given to us. Welcome back. In today's session, we are going to discuss the possible questions from Mendelian Genetics. That is from the sessions that I have so far covered in the previous videos. So let us have a recap on that first. The sessions covered under Mendelian Genetics. The meaning of the term genetics, inheritance, variation. Then we dealt with Mendel's experiments. There we have seen regarding monohybrid cross, dihybrid cross. Then we came across the three laws framed by Gregor Mendel. Then we learned about the concepts like Punnett square, test cross as well. So based on these aspects, I have framed a few questions, possible questions that can be asked for the CBSE board examination or any competitive examination. And I hope it will be useful for you all. But before going into the questions, let me suggest you to subscribe my channel so that you won't miss any sessions that I publish, and also you will have a clear glance of what I have already taken. So subscribe it so that it will be useful for you. Now, regarding the questions, let me start with some direct and easy questions first. Since it's a science subject, almost a question will be there in the question paper starting from the word define. So naturally, definition of the terms that we had already discussed will be there. Let me take you towards one by one. That's like a recap. So first term, define the term genetics. You all might be knowing, isn't it? Genetics, the term genetics, it deals with inheritance and variation. So genetics is a branch of biology which deals with inheritance and variation. Now next term that can be asked under definition is inheritance itself. So what is inheritance? Inheritance, it is a process of passing on characters from parents to progeny. So it's a process. Next, as we have dealt with Mendelian genetics, naturally two terms will be there: monohybrid cross and dihybrid cross. So monohybrid cross means the cross in which the characters differ. The plants differ, the parents differ only in one character. Dihybrid cross in which the parents differ in two characters. So like that, monohybrid cross and dihybrid cross. Then we learned some terms like new terms, homozygous, heterozygous and all. So homozygous, individual means individual or in which the alleles are same. Alleles for both the alleles for a character are same. Such individuals are called homozygous alleles. Individuals, sorry. Heterozygous means individuals in which the alleles for a character are different. One allele will be dominant, another will be recessive. In such a case, you call that character as heterozygous character or heterozygous combination of alleles or heterozygous genes, or that individual as a heterozygote. So homozygous, heterozygous. Same alleles, different alleles. Next term is allele itself. This is a very new term. Alleles are different forms of a gene. Alleles refers to, can be defined as different forms of a gene. Okay. Now what is gene then? Gene is the unit of inheritance. So these are the different terms. Next we came across a term called as test cross. Test cross, as the name suggests, test cross is a cross used to test the genotype of the progeny by crossing it with the recessive parent. So these two terms should be there in the answer. First thing, to test the genotype of the progeny. Next thing, the progeny is crossed with the recessive parent. So these two things should come when you write the definition of the term test cross. Next is regarding Punnett square. Punnett square is a graphical representation which represents the probable genotypes of a cross, genetic cross. So that's the definition of Punnett square. Then we learned two new terms, another new terms, phenotype and genotype. Phenotype refers to the external appearance of the organism, whereas genotype refers to the allelic combination of the organism. So phenotype, genotype. So definitions will be surely asked in the question paper. These are the possible terms that can be asked under the topics which I have covered so far. Next question, next kind of question will be like differentiate between. Differentiate between is what we have learned here itself, but it can be asked like differentiate between. So whenever you get question differentiate between, for example, I am putting a question differentiate between monohybrid cross and dihybrid cross. So whenever you get a question like differentiate between, you have to put it in a column. You have to put it in box and write the two terms monohybrid cross, dihybrid cross on either side and write the difference one by one. Don't write as paragraphs. Put it as points, put it inside the box so that it is presentable and it will be worth valuing as well. Okay, so like differentiate between again can be asked for monohybrid, dihybrid cross. It can be asked for phenotype and genotype. It can also be asked for the difference between alleles and genes or homozygous and heterozygous individual. So whatever we have covered under definition, some terms can be picked up from there and can be asked under the differentiate between session also. Okay. Now, so those were some direct and easy questions that I had put forward to you. Then some long answer questions will be, it will be also direct. State Mendel's law. State the three laws of Mendel. When you state those three laws of Mendel, you should not bring any change in the sentence. You have to state the laws as I had taught you in the previous sessions. You cannot frame your own laws and put it into your own sentences. You cannot change the sentence. Please frame the law as it is. Okay? And do it well for the examination. Then questions can be asked from what is the inference? Inference between F1 and F2 generation. What inference did Mendel get from monohybrid cross F1? Monohybrid cross F1. He got all tall, isn't it? What inference did he get in F2? Some inference means what result did he get in F2? Some were tall, some were dwarf. So from this F1 and F2 he inferred that some characters are expressed, some are suppressed. So from that he could infer the concept of dominance. Okay. Then what about from the dihybrid cross? What did he infer from the dihybrid cross? He inferred that the factors, he told those genes, our genes as factors, right? Those factors separate out independently. That means factor for a character is not depending upon factor for another character. You remember, children? Round seed can be yellow in color or green in color. That means seed color and seed shape are the two characters that is not depending upon each other. So alleles separate out independently. So these were the inference questions that can be asked from the chat portion. Then from last year onwards, CBSE has introduced many multiple choice questions that will be useful for many competitive exams as well. Okay, so those multiple choice questions, I'll just put one by one for you. So before going into the multiple choice question, let me explain this type of a question. Here, this is a type of cross. This is there in the textbook as well. You can see here, tall plant with yellow seed. So what is the phenotype? Plant is tall, having yellow seed. And what is the genotype? Capital T, small t, capital Y, small y. So phenotype and genotype is given in the question paper. Tall plant with yellow seed is crossed with tall plant with green seed. Both phenotype and genotypes are given. What is the question? What proportion of the phenotype, we have to determine only the phenotype, is expected to be tall and green, dwarf and green? So two questions they are asking. In the progeny that you get, how many numbers will be tall green and how many will be dwarf green, or how much percentage will be tall green and how much percentage will be dwarf green. This is the question. Let us try to find out. So we have to write a cross here, simple cross. 2 characters are taken, dihybrid cross. So what is the parent here? Tall plant with yellow seeds crossed with tall plant with green seeds. Genotype is already given, that is what is quoted. Here also genotype is given. So these are the parents. Next step is what? You have to frame the gametes. So since this is a dihybrid cross, it will be better to express this in Punnett square. Let me first write the gametes. Capital T, small t separated. Capital Y, small y. Again, capital T can combine with small y. Small t can combine with capital Y. So you can get here 4 different combinations. Okay. Now what about this side? Here this allele for seed color is same. So what you have to separate is allele for plant height. Capital T, small t separated. Now small y, small y. Even if I write once again, I will get the same thing. Capital T, small y, small t, small y only. So I am writing here only once. Okay. So these are the gametes. Now to get the progenies correctly, what should I do? I should write it in the Punnett square. I should write it in the Punnett square. Okay. So I'm just going to draw the Punnett square. Punnett square. For that, I have to write the gametes on the left and on the top. Okay, first let me write the gametes on the left. Capital T, capital Y, capital T, small y, small t, capital Y, small t, small y. I have written the gametes. Now next gametes here. Capital T, small y, capital T, small t, small y. 2 gametes only. Now next step is separate out the gametes. Gametes separated. Make it as a square. Okay. Squares are formed. Now next, fill up the squares. How to fill up the squares? Left and top gametes you take. So capital T, capital Y, capital T, small y. What is this phenotype? We have to determine the phenotype. How many tall green we have to determine. Okay. And how many dwarf green. So tall green how many? Next is dwarf green how many? Tall green how many? Dwarf green how many? Okay. This is tall, but is it green? No, because capital Y is there. So we need not take that. Next column you take capital T, small y, capital T, small y. This is tall and also green. So I am getting tall green 1. I am writing here 1. Tall green 1 I got. Next, small t, capital Y, capital T, small y. Oh, here capital Y is coming. So it is not coming under green, so I can ignore this. Next one, small t, small y, capital T, small y. Both are small y's. So it is green and capital T is there, so it is tall. So it is tall green, 1 more I got. So I am writing here. So tall green I got here, tall green I got here. 2 columns. Okay. Now here again capital T, capital Y, small t, small y. Okay, I am just writing the ideals. This is tall yellow. I cannot write. Next one, capital T, small y, small t, small y. This is again tall green. So I got tall green 1 more. 3 I got. Next, small t, capital Y, small t, small y. Okay, so this is dwarf but yellow. So I cannot take this. Last one, small t, small y, small t, small y. This is dwarf as well as green. So I can take this also 1. So how many total here? Progeny 1, 2, 3, 4, 5, 6, 7, 8. Out of 8 progenies, I got 3 tall green and 1 dwarf green. So I can write here 1 by 8 is dwarf green, and instead of this 1 plus 1, 1, I can write 3 by 8 is tall green. If I have to convert this into percentage, how can I convert this into percentage? Just multiply by 100. 3 by 8 into 100. How much will you get? You'll get some 37.5. Yeah, 37.5. Similarly, 1 by 8. You convert this into 100, multiply by 100. Convert this into percentage, okay? Then you will get here 12.5 something. You can work it out, check it out, okay? So here, if question like this is asked, what proportion, if such a word is coming, what proportion, it is better that you express it in the form of percentage. Just whatever number you are getting here, divided by the total number of progeny into 100. Okay, so that is how you can solve such a question. So here in this question I have given you as an example, whatever the genotype is given here, don't worry, frame the gametes first, put it in the Punnett square. Whatever the phenotype is asked, you write that here one by one, and as and how you get, you keep on adding here number 1, 2, 3, so that you won't miss at the end and you won't get any confusion sitting in the examination hall. Okay, so it will be easy for you, and then at last you multiply that by 100 so that you can express it in percentage also. So whatever phenotype is asked, you bother only about that one, need not worry about the other things. So hope such questions, if it is coming, you can solve it with ease. Okay. Now, as I've told you, questions can also be of multiple choice forms. Multiple choice forms, and some of the choices given will be a bit confusing. But if you are really thorough with the concept, it will be very easy for you to solve it out with the keywords that we have learned in the previous session. So that's why I have told you in the beginning itself, please subscribe the channel so that you will come to know each of the videos systematically. You can play it out, and also you will not miss any future videos that I am putting, because I intend to put the things in a systematic manner, one by one, then take a revision, then frame questions based on that, and then move forward. So if you have any suggestions in this regard, or any doubts in the answers, or if you want more questions to be discussed, please approach through my channel. Okay. Now multiple choice questions, as I have promised. You see, a question is given that a cross between tall plants. The question is like this: a cross between tall plants, tall plant crossed with tall plant. A cross between tall plant resulted in a few dwarf offsprings, not all, a few dwarf offsprings. What would be the probable genotype of the parent? This is the question. What we have to find out? The probable genotype of the parent. So, cross between two tall plants giving some dwarf plant, we have to find out the genotype of the parent. Okay, so we are getting what plant? Dwarf plant. So in the genotype of the parent, at least one should be small t. So we don't know both the parents, but we are writing here small t for both the parents. Okay, but both the parents is of what type? They both are tall. So naturally both of them should have capital T, capital T. Only then they can be tall. But the progeny is coming dwarf. So in them the recessive allele small t also should be there. If I write like this, capital T, capital T, capital T, small t. In this case also, both the parents are tall and there is one allele dwarf. Take this case. We will take as two cases. One allele is dwarf. What will be the gamete here? Capital T. Here the gametes will be capital T, small t. If you cross this, what will you get here? Tall. If you cross this also, you will get here tall. But is our expected thing coming here? Dwarf progeny is coming here? No, it's not coming. So I cannot take the parent like this, isn't it? Both are tall, but I cannot take here both homozygous tall. So I have to change the genotype. Capital T crossed with capital T, small t. So here the gamete is coming like this, capital T, small t. Here also capital T, small t. So I can get, as we got before, three tall parents and one dwarf. So tall and dwarf. So question is, I am getting a few dwarf progeny. A few dwarf progeny I am getting. What is the genotype of the parent? So genotype of the parent is, parents are heterozygous. You should not write the answer as genotype of the parent is capital T, small t. Don't write the answer like this. You should write the answer, genotype of the parent is heterozygous. That is the answer. But if in the multiple choice the parents combination is given like this, capital T, like this combination are given, then you choose that and write. Otherwise, if simply it's a question, what is the probable genotype of the parent, you should write the word heterozygous. So genotype is asked, write the word heterozygous, homozygous likewise. So this is clear to you. So this is the probable genotype of the parent. Parents are heterozygous tall. Okay. Now, so this is one multiple choice question which can be asked. So you can write this very easily as well. Just work out. It will not take half a minute. Work out and choose the correct option. Now, some other question can be asked, like in the F2 generation, what is the number of phenotypes? Different phenotypes you are getting, different types of phenotypes. Okay, what is the number of phenotypes? And then also number of genotypes. How will you make this out? Number of phenotypes and number of genotypes. See, phenotype we are getting the ratio, no? 9:3:3:1. That means homozygous tall. This is having both dominant form, one both recessive form. Here one form will be dominant, another recessive. Here the vice versa. So you are getting 4 combinations, 4 types of phenotypes. So the number of phenotypes in F2 generation is 4. Number of genotype is 9. How will you get this 9? Dihybrid cross. You take each one of the genotype and count. You will get there 9 different types of genotypes. Okay. So this is a question. Number of phenotypes in F2 generation for a dihybrid cross is 4 and genotype is 9. Now, I am giving you, next question, okay, I am giving you here 4 choices. This is one, second one, then third one, and fourth one, like this. Among these four choices that I have given you, which one is heterozygous? Which among the following shows a heterozygous organism for 2 characters? Diploid heterozygous organism. Here you see both the alleles are same. This is not the answer. This one, alleles are different. Here also alleles are different. So this could be the answer. Put a point there. Next one, this is same. So this cannot be. Here these two are same. So this cannot be. So we are sure that this is the choice. Choice for which one? This choice number 2 represents that the combination of alleles purely points towards heterozygote genotype. Okay. Now, another question can be like, what type of cross or with which parent will you cross the progeny to find out whether it is homozygous or heterozygous? So the question is, with which parent will you cross the progeny to find out the genotype of the progeny? Yes, so our mind should go towards test cross. Test cross is used to determine the genotype, isn't it? So what is test cross? Cross with the recessive parent. In the definition itself it is there. So to test the genotype of the progeny, you cross the progeny with recessive parent. So answer is recessive parent. Now children, last but a little bit question to be thought of, little bit. But I'll give you an easy formula to find out that for the exam point of view. Anyway, this is there in the NCERT textbook as well. So, this is a question. A diploid organism is heterozygous for 4 loci. I'll tell you what this loci and all is. For 4 loci, how many gametes can be produced? How many gametes can be produced from it? So the question is, an organism is heterozygous. Heterozygous means the alleles are different. Alleles are different for 4 loci. The word loci, this is a plural form of the word locus. Locus you can understand like the word location. Location. What is a location? Location here means location of the alleles on the chromosome. I'm just representing a chromosome like this. On chromosomes there are certain regions on chromosomes where alleles are present. Those regions are called as locus or loci, plural loci. Okay. So here the question is a diploid organism. Diploid organism means organism will have 2 sets of chromosome, one from his father and one from mother. So 2 sets of chromosome organism is having homologous pairs. That is, organism is diploid. And for how many loci? 4 loci. Here we have taken 4 loci. Just 2 chromosomes, 4 different locations. So I am just marking it like this. 2 chromosomes, I am representing it with the help of a line. And I am marking the 4 locations. 4 locations, I am putting it as A, B, C and D. 4 locations. So same will be there on the other chromosome also. That is why the organism is called diploid. 2 sets of chromosome is there. But are the alleles same? No, it is different because the word heterozygous is given. Okay. So here I am putting the recessive form, small a, small b, small c and small d. Now the question is, how many gametes can be produced? So what I have represented here is the statement. Diploid organism, so I have taken 2 chromosomes. It is heterozygous for 4 loci, so 4 positions I have taken and 2 different dominant allele and recessive allele I have represented. Question is, how many gametes can be produced? So for that, first take up the case of single allele combination, capital T, small t. This is heterozygous for 1 loci. So how many gametes are produced here? 1 capital T, 1 small t separated. This is one case. If I had taken organisms which are heterozygous for 2 loci. 2 loci means dihybrid cross there, okay. Capital R, capital Y, small r, small y. These are, they change in 2 different alleles are there. So here 2 different alleles are changing. So how many gametes I am getting here? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So here I am getting 4 gametes, right? With one heterozygous condition, I am getting 2 gametes. With 2 heterozygous condition, I am getting 4 gametes. So with 3 heterozygous condition, how much will I get? I should use the formula 2 raised to n, where n shows the number of heterozygous loci. Okay, number of 2 raised to n. Remember this formula 2 raised to n, where n is number of heterozygous loci. This is very important. N number of heterozygous loci. Just put the number here, you will get the gamete. This will give you the number of gametes formed. So here 1 heterozygous loci, 2 raised to 1, so 2 gametes. Here 2 heterozygous loci, 2 raised to 2, so 4 gametes. Here we are having 4 heterozygous loci. So for 4 heterozygous loci, how can I write? 2 raised to 4, that is 2 into 2 into 2 into 2. So how much? 2 into 2, 4, 4 into 2, 8, 8 into 2, 16 gametes I will get. This is case only when the genes are not linked. Okay, this is the case when the genes are not linked. I am getting how many gametes? Probably I will be getting 16. This is a tricky question that can be asked, but it can be easily solved within 30 seconds. You can solve this if you know this formula 2 raised to n, where n shows the number of heterozygous locus. So this is true only when the genes in this heterozygous loci are not linked. If they are linked, the number of gametes it will reduce. Okay, so what is that linked gene and all will be dealt in the coming sessions. So in the coming session we will be taking another concept, linkage, crossing over, and also some concepts which is differing from the Mendelian principles. Mendelian principles are not universal. Some plants, some animals, they differ in that. So we will look into all those things in the coming session. Thank you for now.